Hello and welcome to this presentation. I'm Suman Chakravarti from Multicase Incorporated, and today I'll be talking about different computational approaches that we are working on in order to predict carcinogenic potency of untested nitrosamines. So nitrosamines are represented by the general structural formula as you can see on the screen, and they are known as potent carcinogens in animals as they have been tested in rodent models, and therefore they are reasonably anticipated as human carcinogens, and they are in the cohort of concern group. They are found widely throughout the environment that we live in air, water, food, tobacco smoke, cosmetics, and most importantly as impurities in drugs. They came to our attention in recent few years by several high-profile recalls of medications as they found to contain unacceptably high levels of nitrosamine impurities. One of the important aspects of the toxicity of nitrosamines is that many nitrosamines need to be activated metabolically to become DNA reactive. And their metabolic scheme in general is shown in this slide. And as you can see in the last step, there is a formation of a DNA adduct via the formation of a diazonium ion. And in their metabolism, the important step is the alpha hydroxylation, and this alpha hydroxylation step is affected by electronic as well as steric structural features at the nitrosamine center, and these factors will become really important when we'll be developing different types of computational approaches to assess the potency of the nitrosamines. So the nitrosamines can form in a variety of ways, but in general, whenever there is a presence of a nitrite and an amine, there is a chance of formation of nitrosamines. So in the environment, they can form via combustion, in the drinking water from the disinfection processes and from the industrial contamination. In food, they can form in meat, fish, and other products at high temperatures between the reaction of nitrite and amines. In the medicine, they can form in the synthetic processes, for example in the synthesis of tetrazole rings, or they can form from the raw materials by the reaction between nitrites and the secondary or tertiary amines at acidic conditions. So in this slide I have shown few examples of drugs that were found to contain nitrosamines as impurities and were recalled. And the issue, as far as I remember, started with the antihypertensive drug called valsartan, or in general they are called as sartans. But the nitrosamines were also found to be present in other types of drugs like proglitazone, aminophenazone, amitriptyline, ranitidine, and other types of drugs. The latest example is the drug called irbesartan, which was recalled just a few days back. And one thing is common in these drugs. They contain an amine either in their some steps in their synthesis, or an amine is present directly in their structures. So before we go into the discussion of any kind of computational approaches for nitrosamines, let's talk about what are the currently available toxicological assays for the toxic effects of nitrosamines. And there are two assays that comes to my mind. One is the Ames mutagenicity assay and the 2-year rodent carcinogenicity assay. The Ames test is an in vitro test which measures DNA reactivity in terms of bacterial mutated colonies in the bacterial cultures, either in Salmonella or in E. coli. But it doesn't measure really well the carcinogenic potency. It just tells you if the nitrosamine or any other compound that is tested in this particular assay is going to be mutagenic or not. However, the rodent carcinogenicity assay gives a good indication of the carcinogenic potency. It tells the amount of dose that is required to produce tumors in half of the population, at least in the rodents, the nature of the tumors that are formed, and the organs that are involved. But this assay, as you can imagine, is difficult to perform. It requires a large number of rats or mice or other rodents. It is expensive and it takes time to finish. There are several issues which are associated with the assessment of carcinogenic potency of untested nitrosamines. If we are trying to build a computational approach, the availability of data from the experiments is very limited. If we're lucky, then we can find about 140 nitrosamines for which carcinogenic potency data is available from CPDB database or from literature. And the characterization and ranking of the carcinogenic potency based on animal data is difficult. If we are talking about Ames test, mutagenicity test, this is a good test for finding out if a compound is going to be carcinogenic in rodent or not, which is a qualitative measure, but it is not going to be predictive enough for finding out the potency of the nitrosamine in the humans. The issue of metabolic activation to make the nitrosamine toxic is another factor that pose additional challenges, because the metabolic systems in rodents and the S9 metabolic activation that are used in the Ames test is also a complex and difficult step that would pose challenge when we are trying to translate the effects of the nitrosamine from rodent or bacteria to humans. So now let's talk about the main theme of this presentation, and that is the development of different types of computational approaches to predict the potency of an untested nitrosamine. So various techniques come to my mind, and the first one is a quantitative technique, which is quantitative structure-activity relationship or QSAR models that can predict the potency in terms of TD50 values directly. There are qualitative techniques which are known as SAR techniques. They can identify various structural features in the nitrosamine molecule that can either make a nitrosamine more potent or less potent, and these techniques are qualitative in nature. Other techniques include the analog or surrogate chemical based approaches, and these chemicals are similar chemicals to the query chemical, and they best represent the steric and electronic factors that affect the potency of the query nitrosamines. And I'll be talking about these approaches more in more detail in the later slides. Approaches that can compute the speed or degree of metabolism in the untested nitrosamine can also be very helpful, and I'll be touching on those briefly in the upcoming slides. The final technique that I can think of is the quantum mechanical calculations that can compute the reactivity of the query nitrosamines. So these are a few techniques that we can think of in attacking this problem of nitrosamines. There are quite a few challenges that are associated specifically in terms of the computational approaches for nitrosamines. And the first one is obviously the availability of limited amount of experimental carcinogenicity data for nitrosamines. The other difficult problem is that all the training data that we can get our hands on is for small nitrosamines. So these nitrosamines may not be representative enough for the kind of nitrosamine that are present in terms of drug impurities that we have already seen or we are going to see in the future. And if we use this data for small nitrosamines to build QSARs or other types of computational approaches, they may not be predictive enough for the complex nitrosamines. Also, the carcinogenicity data, the available carcinogenicity data, not all the data is robust enough to build computational approaches. Now let's talk about the different possible computational approaches that we can take advantage of in the nitrosamine issue. And the first one is the quantitative structure activity relationship technique, or QSAR techniques. So QSAR modeling is a empirical mathematical modeling technique which relates the change in the molecular structure with the change in their biological activity. And the first step in building a QSAR model is the transformation of the molecular structures to so-called descriptors, or X variables as they are called in mathematical terms, and then using some sort of mathematical or statistical technique to relate these X variables with the response variable, or the Y variable, which is nothing but an experimentally measured property, biological activity, or a calculated property. So basically this is a correlative technique. Now the QSAR models has been proven to be very or extremely useful in various fields like drug discovery or predictive toxicology or in property predictions, because they have allowed us to use correlative modeling just by using a set of examples of molecular structures and their biological properties instead of computing these properties directly from the molecular structures. Another closely related technique or computational technique is the structure activity relationship, or SAR methodology. This technique is qualitative in nature, that is, they can only predict in terms of qualitative values, high or low, something like that. And they are usually composed of structure activity rules that are created by human experts. And these rules are often associated or decorated with biological or mechanistic or chemical mechanistic explanations, and that makes it more interpretable, highly friendly to the toxicologist or the chemists, and they are very popular in the predictive toxicology field. Now in this particular slide I have shown that even in the case of nitrosamines we can make very simple rules that show the basic trends in the carcinogenic potency of nitrosamines. For example, if a nitrosamine is flanked with small alkyl chains which are unsubstituted at the alpha carbons, usually these kinds of nitrosamines are highly potent. On the other hand, if the alpha carbon is substituted, they become less potent, or if the alkyl chains are long at both ends, still they are less potent. So these kind of observations can be coded in computer programming languages in small rules, and they can be used to build a small system. And these coded rules can then be transformed into a computer program to be used for prediction of unseen and untested nitrosamines. And to make these rules more sophisticated, we can keep on adding more and more rules that we can think of or the experts suggest us, and gradually it can become a sophisticated technique to predict potency of untested nitrosamines. So this is basically the structure activity relationship technique, or the SAR technique. I'd like to talk about another computational approach which are called analog or surrogate based methods. And these methods are based on finding chemical compounds that are similar to the query compound. So the analogs are compounds that have similar structural features or similar physical, chemical, or biological properties. Now these analogs can be searched by using the principles of chemical similarity, and the chemicals are called similar if they share same or similar structural features, and if the majority of the structural features are common between two chemicals, we can call them very similar, and if no or very few features are same, we can call them dissimilar compounds. And once we made a search for a particular query compound, then we can make a prediction based on the properties or biological activity of few closest or few very similar analogs. So the basic assumption here is that similar chemicals share similar biological or physical chemical properties. Now the example that I shown here in this slide, that is not exactly the nitrosamine problem, but this is an example of using an analog based approach to find acute oral toxicity of a particular query molecule, and the query molecule is shown on the top, and the few very similar analogs are shown at the bottom part. 5 similar analogs were found, and the final predicted value or the log LD50 of the query chemical, predicted LD50 of the query chemical, is nothing but simply the average of those 5 average experimental values, experimental LD50 values of those 5 analogs. So that is the principle, and we will see in the upcoming slides that we can use a similar approach to find analogs for an untested nitrosamine, and that we can use to predict its properties. In the next few upcoming slides, let me talk a little bit in more detail about using the analog or surrogate based methodologies to predict properties of untested nitrosamines. So the analog based approaches are not new in computational toxicity, and they are widely used for estimating or predicting properties of compounds for which either there are no experimental data available or the data available is not adequate enough. And in terms of QSAR predictions, analogs are commonly used to either support the predictions or to refute the outcomes. Now, in the case of ICHM7 type of QSAR analysis of mutagenic impurities, analogs are commonly used to find out or to validate the relevance of identified alerts or structural alerts in mutagenic impurities. So once an alert is identified in a compound, we can find some closely similar analogs, and by reviewing them and checking their experimental aims data, we can find out if the alert is relevant or not. In addition, they are commonly used in boosting the confidence of in silico predictions by resolving equivocal or inconclusive results, or sometimes they are also successfully used to resolve out of domain outcomes from in silico predictions. So in summary, analog based approaches are a tried and true method and that are commonly used in computational toxicity approaches. So in the last few slides, I talked about the concept of chemical similarity in trying to find similar chemicals to a particular query chemical. But there is another concept that is important, and that is the concept of local similarity. So local similarity is the measurement of similarity when we only consider structural features around a particular alert, and the concept of global similarity is when we consider the whole molecule in the measurement of chemical similarity. But when the DNA reactivity is the main responsible factor for a toxic property like mutagenicity or carcinogenicity, we have seen that global similarity measurements do not always give the best analogs. On the other hand, if we use local similarity, then the analogs that we find are relevant, more relevant, and they give us better predictions. And we have seen this effect when we try to find analogs in the case of mutagenic impurities. The example that I'm showing here is trying to show the same concept. That is, the query chemical is an aromatic amine, but the amine is attached to a thiodiazole ring. So if we find few analogs that contains the same ring, disregarding the rest of the chemical structure, we are getting Ames negative compounds or the Ames negative analogs. So on the basis of just these 3 analogs, we can, with some degree of confidence, we can say that when aromatic amine is connected to this kind of thiodiazole ring, they are usually negative. And as you can see, the last analog is much bigger as compared to the rest two analogs. So if we measure the global similarity, the last compound would give very low similarity. But if we focus only on the local similarity, all these three chemicals have very high degree of similarity to the query compound. So we found this type of measurement of similarity is very useful, and we published the results a few years ago in a journal. So we'll be using this concept in determining the properties of nitrosamine in this particular work. Since we are talking about the local similarity approach, it is worth mentioning few factors that are important in a nitrosamine structural environment. And there are a few factors that comes to my mind, and the first one is the degree of substitution around the nitrosamine functionality and the effect of steric hindrance, particularly steric hindrance that are present at the alpha position to the nitrosamine group. And these steric hindrance usually act like mitigating features, and they reduce the carcinogenic potency. The effect of electron withdrawing or electron donating group should also be taken into consideration. But besides all these local features, there are few global properties that should not be forgotten. And few of them are molecular weight or physicochemical properties like water solubility. And these global properties usually affect the availability of the nitrosamine at the site of toxicity. In this slide, I would like to show you another computational approach which is related and relevant in this problem, because as I have talked about that many nitrosamines need to be activated metabolically to exert their toxic effects, carcinogenic effects. So a related computational approach that we have used in an entirely different program in which we try to predict the metabolism of xenobiotics. And the computational approach tries to find out the reactivity of a site of metabolism. And the main concept is that the reactivity of a site of metabolism depends largely on the structural environment around it. So the picture that I'm showing here is not for nitrosamine, but the effect of structural environment on the site of an aromatic hydroxylation. And the approach records different atom types at certain distances from the site of metabolism and then factors those things in and then uses them to compute if that particular atom that is shown by the red dot here would undergo a metabolic transformation or not. So as I'll show you in the upcoming slides, that we can keep this approach in mind and try to use the same concept in calculating the local similarity of nitrosamines. So in this slide, I'm showing the use of the same concept of predicting the reactivity of site of metabolism, but now using a specific nitrosamine. So this nitrosamine was inputted into our MetaUltra software to predict its metabolites, and as you can see that the program was able to give us the DNA reactive species. But I'm just trying to show you this process of predicting that metabolite. So the orange dot is the site of metabolism, specifically the site of alpha hydroxylation, and the gray dots on the bottom part of the slide is showing the different atom types present around this orange dot. And these different atom types, they encode the structural environment, and they are taken into consideration to find out that yes, the site would undergo a hydroxylation successfully. Let's ask a question now. Can you use the same computational principles to search for analogs that we are interested in? And I think the answer is yes, but there are some problems. And one of them is that we are not primarily interested in predicting metabolites for nitrosamines. If we find out that our query nitrosamine is giving us a particular metabolite, that do not tell us directly about its carcinogenic potency. So it is not really useful. The other problem that I think is that the metabolism data for nitrosamine is very limited. When I searched in our metabolism databases, I could not find experimental data where it's telling me that this particular nitrosamine is undergoing alpha hydroxylation to give this particular metabolite. So that data cannot be used effectively in building up computational approaches for predicting metabolites for nitrosamines. The other thing is that the carcinogenic potency of nitrosamines is not only affected by metabolism, but there are other factors that plays important role, like physicochemical properties. So we can keep this computational approach of predicting site of metabolism in our mind, but we cannot use it directly. We have to do some other things to make it more useful in this particular problem. The first computational approach that I tried is to build a simple QSAR model, and the training set was used, which is comprised of 139 nitrosamines with their experimental TD50 values available from the mainly from the CPDB database and from literature. So the training set is comprised of 139 nitrosamines with their experimental rodent carcinogenicity values, and the descriptors that I calculated are nothing but different atom types present around the nitrosamine group at certain bond distance away. So those are the ingredients of the QSAR, and the statistical methodology to build this model was a simple regular regression technique. Now the first step before I built the model was to use a variable selection process, and this variable selection process was an automated process which identified 9 features that are important, and they were identified using a 10-fold automated cross-validation technique. So those features are listed in the table shown on the right side of the slide, and as you can see, the regression coefficients of all these 9 features are all negative. That means they are all mitigating features, that is, they reduce the potency of the nitrosamine functionality. So these regression coefficient can be used to predict the carcinogenic potency of a nitrosamine that is not present in this model. Few things that I would like to note specifically here, and that these features are not handpicked, as I mentioned. They are picked using a automated 10-fold cross-validation, so these features are not expert rule based. So this summary of the QSAR model that I tried as the first technique in order to build QSAR models for nitrosamine carcinogenic potency predictions. So we can use this newly built model, QSAR model, to first predict the carcinogenic potency of the 139 nitrosamines from the training set itself. So that's exactly what is shown in this slide, and that is the predicted TD50 value, or rather negative logarithm of the TD50 values of those 139 nitrosamines. So, and they are sorted based on the predicted value. So in the picture, the black bars are the experimental carcinogenic potency, and the orange bars are the predicted carcinogenic potency by the QSAR model. And when we sort the values based on the predicted values, you can see that the correspondence between the actual experimental values and the predicted values are very decent. And on the left side of the plot are the less potent nitrosamines, and on the right side of the plot the nitrosamines are highly potent. So this plot actually shows that there is a good chance that we can use this QSAR model to predict or to prioritize the nitrosamines in terms of their carcinogenic potency, and there is also an indication that the features around the nitrosamine truly have something to do with their carcinogenic potency, and there is a quantitative correspondence between these two things. So this is the summary of the prediction of the training set nitrosamines using the QSAR model that I just showed one slide away. Now, can we use the same principles of the importance of local features to try to find relevant analogs to predict carcinogenic potency of nitrosamines? So in order to do that, what we need to do is to build a fingerprint, because fingerprint of a chemical is usually the way for searching analogs. Now, as we have already seen that there is a good indication that the local features have quantitative relevance in terms of the potency of the nitrosamines, we can try to encode those features in some form into a fingerprint and use that fingerprint to search for analogs. So, and when I say analogs, that means analogs, nitrosamine analogs, which already have their experimental TD50 values available to us. So, this slide I'm showing here is the effort of trying to build a fingerprint by recording different atom types around the nitrosamine group and put them into bins, and that is essentially the fingerprint. So on the left side of the picture, you can see that I have recorded atom types from one bond distance and built a small fingerprint. Then I went one more bond away from the nitrosamine group and then built another small fingerprint, and then another bond away to create another small fingerprint. So 3 different fingerprints were built by going one bond distance away from the nitrosamine functionality, which is shown by this orange circle on the left side of the picture. So these 3 subset of the fingerprints, which are nothing but recording the counts of different types of atoms around the nitrosamine functionality, can then be concatenated one after another to get the final fingerprint, which is a bigger fingerprint, which records or encodes the structure and environment around the nitrosamine functionality in a mathematical form. And this is just a schematic representation. It is a simplified representation. In actual practice, these fingerprints are much larger. Almost 1,000 elements are present in the fingerprint. And this fingerprint contains the information about the structure and environment around the nitrosamine group, and it can be used to search for analogs for which we have experimental carcinogenicity data. In this slide, I'd like to note few considerations that are to be kept in mind while reviewing the results of such analog search that I just showed. And the first one is the robustness of the experimental data for the analogs. If the experimental data is not robust or not reliable, then obviously that analog cannot be used reliably in the prediction process. The presence of various activating and deactivating features are also to be considered by the expert, and the contributions of various features has to be considered. And contribution means which features are highly mitigating, which features are less mitigating. So if we consider them, then we can get a sense of how potent or less potent the query chemical could be. And few of those contributions I have already noted in the previous slide, where I show the regression coefficient. That gives us a sense that not all the features, mitigating features, are equal in nature. Some are highly effective, some are not that mitigating. Then the ADME and physicochemical properties should also be considered. So for example, if a nitrosamine analog is highly soluble in water, most likely it is going to be flushed out of the body before it can exert its toxic effects. So this is one simple way to say that the physicochemical properties are also important factor. And the last one that I think is important is that for a particular query nitrosamine, we should first see that if any one of the analogs that we got are relevant or not. It is not necessary that every time we make a search for analog, we will get relevant analogs. Sometimes we'll just come up with nothing. All the analogs would not make any sense. They will be with very low similarity, low local similarity, and they will be very different from the query chemical. And in that case, we cannot make a proper judgment about the potency of the query nitrosamine. In this slide, I would like to talk about multi-case software and databases that were used in this particular work. And I would also like to mention here that we have recently developed and introduced a new software called QSAR Flex. And this software is not only specific for nitrosamine, but it also does other QSAR tasks that are highly useful. For example, it can predict acute toxicity, ecotoxicity, besides finding nitrosamines, analogs. And it is specifically suited for making analog search. Different types of physicochemical properties can be calculated with high quality models. There are very sophisticated database search techniques are available within this program. So the main software is the QSAR Flex software that we have used for this nitrosamine problem. Specifically, as far as the database of nitrosamine is concerned, as I mentioned before during the discussion of the QSAR model, that we have used a training set of 139 nitrosamines, and we have curated the data for them, the carcinogenicity data. And in addition to the TD50 values, we also have detailed information for every data point, and that includes what are the details of the carcinogenicity assay, what kind of species was used, doses information, what type of tumors were formed, and so on. So this combination of QSAR Flex as software and the nitrosamine database was used to perform the tasks that I'm discussing here in this presentation. So using all the knowledge that we have learned so far and we have discussed so far in the previous slides, and the technique of building the fingerprint that I have explained, now we can make a search for analog for a nitrosamine query chemical for which we do not have any TD50 or carcinogenicity potency experimental data available. And this is a simple nitrosamine with one side of the nitrosamine functionality there is a ethyl group and a phenyl group is attached on the other side. So I have shown in the table the first 5 analogs that were found, and I have also given the physicochemical properties, predicted physicochemical properties for the query chemical as well as for each analog that we found. And the physicochemical properties are water solubility, log P, and molecular weight. And the analogs are also shown with their local similarity measure and the global similarity measure on the second and third column respectively. So as you can see that the local similarity is always higher than the global similarity values, which makes sense. And the analogs, they also look pretty close to the query chemical. Now After the similarity values, we are reporting molecular weight, which is calculated, of course, and the experimental TD50 values in milligrams per kg for every analog. Please note that these are experimental TD50 values for every analog. And then the last two columns are for predicted log P and water solubility values for the analogs. So these experimental values for the TD50 values are taken from the database that I mentioned before, and the physicochemical properties are calculated computationally by two very high quality models that are present and built inside the QSAR Flex software program. On the rightmost side of the slide, I'm showing the TD50 value for the NDMA, which is a very potent nitrosamine, and just for comparison with the values that we are seeing here, TD50 values that we are seeing here for the analogs. And as you can see, that that we can at least make an assumption that the query nitrosamine would be much less potent as compared to the NDMA, whose TD50 value is 0.096 milligrams per kg. On the other hand, the analogs, the first two analogs, are less potent, and so overall, at least we can take a leap and say that this nitrosamine is definitely going to be somewhat less potent as compared to NDMA. This is another example of searching for analog for a query nitrosamine, but in this case the nitrosamine is much more bigger as compared to the previous one, and this is a reported nitrosamine impurity for desloratadine drug. So here the analogs are not as straightforward as the previous one, and as you can see that the local similarity values are lower, but still the technique was able to find analogs that maintained local similarity as far as the features around the nitrosamine functionality is concerned. At least the first three analogs, they have high quality of similarity, more than 0.6, and the global similarity of course is lower because the query nitrosamine is much bigger as compared to the available analogs. However, as you can see, the last two analogs, which are highlighted with yellow background, they have high water solubility as compared to the first three analogs. So most likely those two analogs are not going to be reliable to be used in this prediction because they will be excreted much faster as compared to the other analogs, and their experimental TD50 value will not be meaningful to be used in this prediction. However, overall, still we can say, based on the TD50 values of the first three analogs, that this nitrosamine, query nitrosamine, is even less potent as compared to the previous example from the previous slide, and its TD50 value is most likely to be even lower, even less potent as compared to NDMA that we are again showing on the right side of the slide. So these two are basically two case studies that we did just to show the promise that analog search are relevant and can be used. So in summary, we can say that the analog search methods, which are based on the principles of local similarity, are potentially useful for carcinogenicity risk assessment of nitrosamines and prioritizing them. The structural environment in the vicinity of the nitrosamine functionality has quantifiable impact on their potency. These features can be modeled in the form of QSAR models to give us quantitative predictions of carcinogenic potency of nitrosamines, and they can also be encoded in the form of fingerprints that can be used to search for good analogs, which will give us another way of risk assessment. So in summary, the use of computational approach has definite potential in terms of the risk assessment of nitrosamines. I cannot end this presentation without thanking and acknowledging the help that we received from individuals to conduct this research. Specifically, this research was conducted under a research collaboration agreement with CEDAR US FDA, and from the same department of US FDA, I would like to specially thank Dr. Naomi Krulak, Dr. Marlene Kim, and Dr. Govindraj Kumaran for valuable suggestions and feedback throughout the project. My colleague from Multicase Incorporated, Dr. Rustam Sayakov, was crucial in data collection and data curation. My colleague Sai Radhamani Allah helped me in the software development in programming. So thanks to all of you. Of course, also a lot of thanks to all of you in the audience for giving me your special attention and time to listen to my talk here. If you have any questions or any queries, please do not hesitate to contact me in the email address that I'm showing here on the slide. So finally, to end this talk, a lot of thanks to all of you.